Once again, and very good morning, all of you, and thank you for joining my session. And during my session, I'll be discussing about the some of the aspects of machine learning. Initially, I wanted to go into the more details of the machine learning and lead optimization approaches. If you want to start with the hit or lead molecule optimization, so what is the key points that we can do? And I wanted to demonstrate that one, but based on my discussion with the doctor, so I came to know that most of the audience are actually new to the machine learning and wanted to start with the machine learning approaches. And the most of the students are with the medchem background. So I composed my presentation with the basics of machine learning and followed by a small demonstration about like how uh, understanding the Jupyter notebook and RT kit, and followed by okay, let's take a problem. How do I address that by using machine learning approaches? That's all about my overview to the today's presentation topics. Is it clear, Doctor? Now my voice?
Yes, clearly.
Yeah, thank you. So before I start with my presentation, I just want to give a overview to the what is Sci Life Sciences and what I do. The Sci Life Sciences is contract research organization, and we offer services to the accelerate discovery, development, and manufacturing of complex and small molecule for the clients. And currently, we are working with a major innovator, small biotechs and pharma companies. And so far, we supported around 100 plus drug discovery projects to the client, and many of the compounds currently in the clinical trials, some of the compounds are already in the market. And coming to the my department, I'm head of the computational chemistry, and I'm taking care of all the cheminformatics and bioinformatic activities, and also I'm my role is also establish AI ML in Sci Life Sciences to support clients for their drug discovery projects and to expedite their project success rate. Right. I think so. Since most of the eminent speakers and they are actually covering this topic, you might be seeing some overlap with other presentations as well. So just bear with me. So before I start with the machine learning model, let's understand what is the drug discovery cycle. I think you might have known this drug discovery cycle. If you want to start with any drug discovery program, so you always start with the basic drug discovery, like basic research about a target. Like for example, I wanted to work on the cancer. So I'll always start with the what is a biological target that is leading to the cancer. Okay, now once after identifying a biological target, then I'll start with the small molecule research or microcycles or followed by covalent or non-covalent inhibitors. Then I'll do everything in the basic research to understand the mechanism, target of action, and small molecule to start with. And once we start done with the basic research and we validate the target, yes, this is the target that is leading to cancer and playing a crucial role, then I'll do the small molecule identification process. It can be hit identification, either traditional drug discovery approaches or computational chemistry approaches or rational approaches. So then once we identify interesting compound, then I'll take a couple of molecules for the preclinical studies, then take the best compounds for the next stage of phase one, phase two, phase three. Then finally, I expect at least if I screen thousands of compounds at this stage, I'll expect at least one compound reach into the FDA approvals, right? Okay. Now, so as per the recent statistics on and each and every drug discovery program, so there are tens of thousands of compounds getting screened in this drug discovery cycle phase and trying to identify the top 10 or top 20 compounds to the next phase. And at least with a backup compound, three to four compound going to the phase one, phase two, phase three, and finally one compound we are expecting with the positive results and the FDA get approved by the FDA and reach into the market. Let's see the same drug discovery cycle into the different funnel. If I see the same flow chart here, now I can see a funnel, right? So I'll take the lakhs of compounds and I'll screen and rank by using a different methods. Either it can be traditional approaches or it can be a computational chemistry approaches, docking or QSA, pharmacophore model, whatever the approach that you are comfortable with that. Then I'll try to screen those lakhs of millions of compounds and try to identify thousands of compounds to screen. Then after screening, I expect at least 10% of the compound success rate move into the next phase. Then so always we are very positive about it, and we expect a very good clinical success rate. Then goes to the next phase, next phase. This entire drug discovery cycle that cost, we are screening around all the possible chemical space that is available in the market, and it takes average 10 years process, but 12 to 15 years on an average cycle, and it costs $2.6 billion, and very very expensive process. So recently I saw an interview of the most eminent pharma founder. Instead of during the interview, one of the uh, um, I think one of the audience asked about if the drug discovery cycle, if that is too expensive, why you are doing a drug discovery? You can start some other business where you can get a profit, right? I can start, the Kingfisher Airlines, or I can start in an Indigo airplane with this cost, right? I can start my own airlines. So, but we are scientists, and we are here to, address the patient needs. That's what the answer. So we are passionate, although there is a failure rate. Okay. So now you are seeing the drawbacks about the drug discovery. There are huge chemical space that need to identify, and the huge filtrations approaches are huge. If your approach like either pharmacophore modeling or traditional approach or bioisosteric approach or docking approach, it has to be like the very well validated model so that you can reduce the failure rate and improve the success rate in the next phase. And you can see it's an average 10 years process, but it might take 12 to 15 years, and the money that is involved 2.6 billion, and the manpower that is involved over the 10 to 15 years. My god, experts from various fields, expert from biology, expert from toxicology, expert from comchem, experts from medchem, to address this issue, right? So can we shorten this? Can we improve the success rate by using a drug improve by existing technologies, right? I'll explain one of the success story from in silico medicine. I think you might be knowing this success story. In silico medicine is one of the AI-based company. They are using a generative modeling approaches to design a compounds. So what they are doing is they here in one of the success story, what they mentioned is within a 46 days they identified, optimized a nanomolar compound, right? And their approach is, let's see the breakdown. The first 7 days they spent on the data set, the reference compound, either it can be clinical candidate or it can be the reference compound from the patents or journals, kinase inhibitors, non-kinase set, allosteric inhibitor, existing protein structures, and IP base and everything. The once the detailed data set and its SPR analysis is done, and the next 12 days they spend most of their time in the generative chemistry by taking the data set of existing compounds against a DDR1 kinase inhibitors, and they model the into generative models. Then they started generating a chemistry, the ideas from the generative chemistry. Then by using a different, rewards and awards, Cohen maps and novelty and pharmacophore hypothesis. And the next 2 days they spent on the prioritization, understanding molecular properties and max fingerprints and clustering diversity, and whether the generated chemistry, whether they are commercially available or non-commercial compounds, they are not available as non-commercially. Then they started with the synthetic chemistry. So what are the compounds that are prioritized from this space? They started analyzing synthetic routes and they started synthesizing them. And the six compound they brought from Uxia. And within this 35 days cycle, they able to synthesize compounds, and they were able to screen the biological activity also. And they finally they come up with a nanomolar compound. This entire activity has been completed in 46 days, and they also identified a nanomolar potent compound, right? So that's what we are seeking, right? So whereas a 10 years cycle here and 12 years of average and $2.6 billion, and here they screened very less number of compounds, which will actually reduce the resources of chemistry, resources of biology, and they screen only from 30,000 ideas to 40 structures. Out of the 40 structures, they come up with a novel IP and most active compound. So you can imagine the resources that they are saving here, and also the number of compounds and the success rate, right? Very interesting, right? Now let's break down the chemistry again, the same workflow in a different way. So traditionally, the hit to lead stage will take 2 to 3 years, right? So here I'm talking about this phase. So traditionally, the second phase will take 2 to 3 years, and if you are failed to identify, or if any company fail to identify a best compound, so they will fail either here or they will fail either here. They have to return, come back to the second stage and do the analysis and do identify the problem, then refine the compound. Again, do the preclinical studies and go into the next phase, right? Rather than failing at the preclinical stage or next stages, we do most of our analysis in this stage. So, on an average, the traditional hit to lead identification stage take approximately 2 to 3 years. But whereas the generative modeling approach, it took less than 2 months, 46 days actually, right? You can see target identification and generative modeling it took around 21 days, and after that, rapid synthesis approach and biology screening, and also they have done metabolic studies and fibrosis selectivity also, and followed by the best compound they selected for in vivo and preclinical test. So all this data is available in a Nature Biotechnology paper, and the resulting compounds they are not only nanomolar potent, and they're also showing a very good PK properties, right? Very interesting, right? And so you can expect now, okay, let's take this approach and apply on my case studies or apply on my drug discovery project. No, you can't do that because the dataset diversity, dataset existing information, and a lot of validation is required to start with that, right? Everything on the machine learning and deep learning approaches is completely dependent on the data, right? If your data points, a number of data points are less, or maybe 100 or 200 compounds, no, you can't start with this approach. You need a maximum number of 5,000, 10,000 compounds which are already tested against this target. Then yes, I can do the deep learning approaches. If the target is novel and if there is no compounds or very less number of chemical diversity, no, this may not be the right approach for you. Whereas here, the in silico medicine has started with a huge data set so that they come up with a, they were able to validate, they were able to generate a best model, and they were able to get the success rate. Any machine learning approaches, either QSPR or QSAR, at least you can start with 100, 200, or 1,000 compounds. But for deep learning, you need maximum data, as much data as you can. The machine learning models and AI models limitation is the data, and not only data, proper data. Because if you see, for example, on the DDR1 kinases, you can get hundreds of patents and hundreds of journals, right? But what is the essay condition? What is what I can say, the biological assay conditions, and what is our activity, like whether they are IC50, EC50, percent inhibition. So you need to clean all this information and use that as an input here, right? And the chemical diversity. For example, the same compound is reported by some journal as active or some journal as inactive. So that is called improper data. Most of the drug discovery having a unstructured data, so you can't simply take it and start it, right? So most of the AIML technologies are very fascinating, but only limitation is data and cleaning the data and selection of the data. Okay, right. So is the machine learning in drug discovery is a new? No, it's not new. You know, the one of the first publication about machine learning methods in the drug target prediction was published in 2018, right? But not actually. So the implementation of neural network and the role of neural network was first published in 1991. So we have been using these machine learning methods in our drug discovery cycle, and only thing that is changing now is open source and the technology and the GPUs, which will actually enable you to handle the huge number of data sets and the algorithms, right? So shall I take a pause? Any questions here, or shall I continue?
So you can continue. Hello? Hello?
Hello.
Yes. Now I can hear.
Oh, okay, sure, doctor. So since it's a quite silent, so I wasn't surprised whether again I lost the connection or not.
Yeah, I think in between you lost connection.
Okay, is it fine now?
Yeah, it's fine now.
Okay, right. Okay, in condition to that, the machine learning applications like in the breakdown if in the drug discovery cycle. So target identification and validation stage. So we'll start with the target prioritization, identification and prioritization by understanding the gene and disease role and druggability and alternate targets. So here in the machine learning, we'll try to understand the metadata of the target and we'll try to understand whether the role of high dimensional data of the target disease and disease association data sets and omics data sets and highly confidence associated information, right? So machine learning also plays a key role in the target identification and validation by using the existing omics data. And compound screening and lead discovery. So we'll start designing the compounds with the desirable properties, whether the compound is having satisfying all the rules that is set by me, right? And you can also understand what is the starting compound and what is the drug properties and everything, right? So large amounts of data is needed. So large amount of data and structured data we need and models for the reaction space and you can define the chemical rules and everything to filter the best compounds and obviously you need a huge data set for to generate ADME models and uh, I think you might be knowing about a deep AI DeepMind where they recently solved protein structures by using AI approaches that is again um, I can say huge developments towards the crystallographics and solving the structure based approaches. And the preclinical development, so we can start analyzing the biomarker data analysis and understanding the cell line data and proteomic and transcriptomic data to understand high quality and quantity data and trying to use that knowledge for the designing itself. And the clinical development, pathology and data sets, the number of image based machine learning models to understand the problems and also improve the clinical development phases, right? So what is the machine learning regular workflow? The first thing is, as I discussed many times, so as I mentioned many times in the previous talk, so first thing is we'll start with a compound database. For example, if you want to start a machine learning model for your target or imagine for solubility or for any ADME properties for absorption, blood-brain barrier and penetration, hERG liability, or genotoxicity, hepatotoxicity. So all the thing is we need a compound data which has to be structured and activity needs to be you know I can say it's a process and cleaned and stereochemistry is addressed, right? All the ligands to be prepared in particular pH conditions, then use that model, use that compound database for the modeling purpose. So here will be first point is a chemical feature extraction. So here the chemical feature extraction is a multiple methods. Let me go back here. So most of the machine learning algorithms take the chemical data as either in a smiles format or chemical chemical fingerprint format, right? I think most of you know the SMILES, simplified molecular input line entry system. So we'll actually provide this SMILES format to the algorithm, right, and convert into a chemical print that contains the information about the molecule, right? There are different fingerprints also available. I think we'll discuss in some other time. And either SMILES or chemical fingerprint, physical chemical properties or potency data are three-dimensional structural properties, interaction fingerprints of graph models and wave models, right? So this is your input for the machine learning models. Okay. So once we convert the database into the chemical feature extraction, either that can be a fingerprint approach or either it can be like the smiles based approach, then we'll and try to understand the chemical diversity of my data set, and then I start with the model learning. Okay, that is all about the machine learning workflow. It's very simple. Okay, if you want to start with the machine learning workflow, like what is the resources that I have? The first thing is, as I mentioned, RTKit is the most well-known Python libraries. If you want to start with that, then deep learning is called DeepChem, and 3D Mole.js or NGL, which will allow you to work on the three-dimensional structures within the Jupyter interface. And when it comes to the interfaces, you have a Jupyter and followed by the pipeline pilot 9. So most of them is our open source except pipeline pilot. So you don't need any additional licensing to start with that. Okay, all you need to have the basic knowledge of Python and understand RTKit and DeepChem libraries and how to work with the Jupyter and start with the modeling, right? And how this pipeline pilot works. So pipeline pilot is very simple. So in pipeline pilot, you have a component, simply drag and drop, and what actually you want to do. Like you need to split your actions, like prepare a data, split the data, process the data, build models, apply model, write the output, right? And you'll have a number of components to do that. Just drag and drop and connect them and define the X and Y variable and generate a model. That's it. And where is the nine? So for NIME, you have a open source nodes and also you have a Chemexa nodes. You have, I think, I believe, Optibrium and also Schrodinger. Everyone supports NIME. Is an open source platform again. So again, you need to define a workflow, like what exactly you want to do. Like for example, I want to import a molecule. Then I want to do the calculate elemental analysis, right? And I want to calculate a accept tone or log p pKa, and I wanted to see a table, right? It's a very Marvin is less coding and simply drag and drop, provide input and define the workflow that what exactly you are planning to do, right? So it's an example screenshot. Now coming to the Jupyter, so is a born out of IPython in 2014. Now Jupyter Notebook is the most are widely used by the data scientist or machine learning scientist, not only on the drug discovery in the various domains. Jupyter Notebook is, I think, one of the best choice for the most of the people over the NIME and Pipeline Pilot, I can say. Or maybe the most of the people consider them because of the exploratory data analysis and their easy catching inbuilt cells and language independent and data visualization model, live interactions with the code and documenting code samples. That is very, fascinating. So I think, as I mentioned, I don't want to bore with too much with the presentations. So that's all about. So far, I covered about the machine learning, what's the role and responsibilities, and success stories. Right? I think we discussed about one of the success story. There are many success stories now, and followed by if you want to start with a machine learning model, what is the basic approach, right? And what is a input variable, and followed by the list of libraries that are available for drug discovery, right? And pipeline pilot 9 and Jupyter notebook, right? So any questions so far? Are we good?
Sir, there is a question from the chat box. How much time it will take to require to screen 1.2 million structures by using machine learning and AI tools?
Okay. So I think Abhijit Debnath, AI and ML methods, uh, uses again what actually you wanted to do that, right? You want to do the docking, or you wanted to do the prioritization or generative chemistry. Based on that, it will vary, right? Hope I'm clarified. So, the Om Prakash Tanwar, is there any open source tool that is available? Yes, as I mentioned, RTKit, which is a Python-based tool, right? I think you can start using RTKit in Python, and that is a completely open source to handle the small molecule libraries. And can you demonstrate how to use the NIME protocols on Chemexon? No, I don't have official license, but I can reach out to someone. Maybe they can get in touch with Dr. Shom Shankar, and they can plan for a demonstration. And but for Jupyter, we need sound knowledge of Python. Yes, definitely, but it is not a tough task. If you spend a couple of months, I think you can start with the right. Even I'm a scientist, but I'm not very good in coding. I was able to learn in few months. Okay, let me demonstrate a Jupyter notebook and come back to you on the questions. Okay, second. Edit this full screen. Yeah. Can you see my screen, doctor?
Yeah, we can see.
Right. So, okay. So before I start with the demonstration, I just want to mention one point. So whatever the data sets or whatever the command that I'm showing here, it is from the open source data, or whatever the model that I'm going to use, whatever the compound that I'm going to use, because of the confidentiality data. I can't show you my own code which I developed, because of the since we are coming from the research organization, and to avoid any further conflicts, I'm just showing a non-confidential data that either that is published or that is available on the public databases. Okay, right. So this is all about Jupyter Notebook. So you can install the Jupyter Notebook from anaconda.org, and it's a very straightforward installation. And uh, followed by, if you before I start, I wanted to advise, if you want to start with a Jupyter Notebook, I advise, there are actually very free courses are available on the Python. First start learning the Python, then come back to the Jupyter Notebook. Okay, then try to understand about the RTKit and deep chem libraries and machine learning modeling, then you can start with the ML. But I don't think you need a 1 year course for this machine learning modeling. Hardly, I think 2 to 3 months of your time is very good enough to start with that. Okay, but I'm not going to the too much coding now, because most of you are from the medicinal chemistry background. Let me give you the flavor accordingly. Okay, so I'll start with the importing Python libraries from the RDKit. RDKit is one of the open source and extensively developing on the cheminformatics packages. So here what I'm doing is I'm just importing RDKit here. From RDKit, I'm importing a different libraries to calculate, to handle the chemistry data, and to handle the physical drawing the capabilities, and calculate physical chemical parameters. Right. All this is to support my activity here. Okay. Now once I import RDKit and the supporting associated packages, so first thing is, I think most of you know about smiles, right? The 1D string of the molecule. So here I'm defining a variable called, that's called molecule one, and from the chemistry library, I can see here R ticket import chem. This is my chemistry library. From this chemistry library, I'm actually calling create a molecule from smiles, right? And this is my smiles, okay? And mol 1. Let me type it for you, okay? Mol from smiles. You can add, let me take the same molecule from the other column, okay? That's it. Then I want to see what is inside the mol 1. This is my molecule, right? Okay. So I was able to read the smiles and I was able to convert into a molecule. Okay. Now on the second step, let's add a hydrogens. Okay. I'll remove to avoid any confusion. Okay. So to add the hydrogens, I'm actually defining another variable called mole underscore example H. Okay. And again, I'm using a chemistry library to do that. Chem dot add hydrogens to the mole 1. Okay. And let's print it. Let's see what is inside. So previously this is my molecule. After adding the hydrogens, now this is this one, right? Okay. Now imagine that is if you are reading a one smiles. If you want to read a SD file, I think you might be knowing that SD file you can create from ChemDraw. So if you have multiple molecules, so generally we used to save either .smi or .sd file. Okay, SD file is the structured data file. Okay, so here I'm defining a variable called w1, and again I'm using a chemistry libraries components from the RDKit and SD writer. Okay, if you want to read the SD reader, so you have SD reader as well. Okay, so please let me know if it is confusing. I can pause and I can re-explain for you. Okay, that is about a file handling systems. Let me take a two compounds here, mol 1, mol 2 again here, right? Let's see what is the mol 1 and mol 2 cation. The mol 1 is this one, toluene, and mol 2 is nothing but of the chlorobenzene, right? And I want to calculate a fingerprint for this one, similarity. Okay, I'm just taking a very basic example. So here what I did was molecule 1, I took a toluene, and molecule 2 is a chlorobenzene. By seeing, you can say, yes, these are similar, right? I just want to give a brief calculation how it does. So here I'm using a allchem library from RTKit, which I already imported here. Okay, again RTKit, right? Hello, I think looks like there is a question. Shall I continue?
Yeah, you can please.
Okay, so there are two molecules here. Okay, now this is the molecule final structures, and here I'm using allchem libraries and using a Morgan fingerprint, and I'm calculating a fingerprint here just by clicking this, either run button. Now let's calculate a tiny motor similarity between the fingerprint of first molecule and fingerprint of second molecule. As I mentioned, I don't want to scare you with the Python code. I'm just very explaining in a very layman terms, okay, so that you can start with the RDKit rather than getting worried too much about RDKit. Right now, the Morgan fingerprint, the similarity is 0.53. Okay, there are different fingerprints. So here just for demonstration, I use the Morgan fingerprint. Okay, now imagine you have smiles or IUPAC names are followed by information which is in Excel or .csv file format. How to read that? Okay, so here I'm using a libraries called Pandas, NumPy. Pandas mostly to handle the file systems, and NumPy to handle the mathematical functions, right? So I defined a variable called df, and pandas I imported as a PD. So I'm using a pandas code here, pandas dot read underscore CSV, define the path of the file, right? So CSV, I think you might be knowing, comma separated file. So what is a separator? Whether space or comma or semicolon, you can define that. So it's a semicolon. Now let's read top 5 headings here. So this is my CSV file. Okay, so which is downloaded from the Campbell. You can see all the molecular weight, smiles, and followed by the, you know, target, IC50, and literature journals and everything. Okay, just let me know if it is bouncing back or if it is too much into the Python. I can switch into the more of Metcam oriented presentation. Okay, now let's see what is the CSV file contains. So CSV file contains the Campbell ID or molecular name and max space, miles, all the essay related field points. It's like CSV is nothing but more of Excel sheet. Okay. Now I want to convert these miles, which is there in this column, into a molecule, right? And let's see those molecules. Now I can see those molecules now. So, I don't need any software. I don't need to install any, uh, what I can say, the huge installations or anything. I'm just by using a Python and RTKit libraries, I'm handling this one. Okay. I was able to read a CSV file or Excel file, which is containing a smiles or IUPAC names. Now I can see those structures now, right? Now let's add those structures into the table itself. Can you see those structures now, right? So I'm not going to the Python commands and how it works and everything. I'm just demonstrating the capabilities here. Okay, since the most of you from the chemistry background. Any questions, please feel free to stop me and ask me before I switch into the next one. Are we good here? Okay, so nice. I have another interesting one. Let's see if it works. Just loading. No, I don't think it is working now. No, it is. This library is not working. Sorry for that. Okay. Okay. Now let's discuss about the Metcalfe point of view. Okay. Now so here coming to the major problem now. So in a chemistry terms, okay, this is my input compound. Actually, client actually provided this compound, and this is my starting compound, and he would like to do a optimization. By seeing the compound itself, only the two concerns that I identify is a walk, right? And another concern that I can identify is a nitro group, right? Okay, so imagine the client is actually saying that the solubility issue or toxicity issue or it is not following the drug-like properties, or it's not within the drug-like properties, or you want to optimize the number of hydrogen bond count or acceptor count or donor count, right? Let's see that. This is my starting compound. So here I use the same RTKit components here. So this is my client compound and the smiles. Just imagine. So I was able to convert into a structure now. Now by using a RTKit, I can able to calculate a descriptors. Let me show you. So here I'm calculating the molecular weight. So I added this compound to a variable called that's target compound. Let's print a molecular weight. So molecular weight of this compound is 362.38. Okay. Now let me calculate the log P. Since the client is saying it's it has a solubility issue. Yes, the solubility log P is a bit high, 1.7, right? The hydrogen bond count is 6 and donor count is 2. Okay. In addition to this one, if I wanted to calculate any properties, so here I can think about like a number of aliphatic rings. Okay. So change the variable and run it. So number of aliphatic rings are 2. Can you see here? It can be a breach or separate aliphatic rings. So this will simply tell me like, okay, this is my molecule and this is the property distribution. The more organized way is, I can see now number of heavy atoms in this provided compound is 26, number of donors are 2, number of acceptors are 6, molecular weight is 362, log P is 1.7. Is this enough to optimize this compound further? No. This will tell me like a profile of this compound. Okay, molecular weight is bit high, or it's okay, and log P is bit high, and number of donors are still you can increase. And by seeing the compound, I can only see that issue with the TBOC here, tertiary butyl and nitro group. But there are many drugs with the TBOC here and many clinical candidates and with the nitro also. Okay, let's say analyze further. I already having a toxicity model, right? So before I start with optimization of this compound, let me go back, switch into the notebook. So here I'm using AntiKit and Matplotlib here. So here I was trying to understand whether is there any issue with my compound, right? So the estimator is nothing but of my machine learning model about the toxicity, okay, and trying to identify the corresponding fingerprints by using a similarity maps, okay. So when I see the whole molecule, the only issue with the TBOC, okay, when I break down this compound into the next stage, let's take only the aromatic component with the nitro group. So the red color indicates the compound, it is having a negative impact on my toxicity. It means that nitro group is a culprit, and also aromatic amine, that is also culprit. You can see the color shade. Now let's break down the other part of the compound. So TBOC again a culprit. So now come back to the same compound. Now I know C-log-P is a concern and the nitro group is a concern. Not only my theoretical knowledge, my machine learning model also able to predict this nitro is a culprit and aromatic nitro and TBOC. So you don't need to address everything all together at once. You can address one by one. And during the applying the machine learning models or optimizing the compound, make sure you are addressing the one compound at a time, one suggestion at a time. Okay. Let me take these miles, go back to the draw here. So most of the scientists, when you see the nitro compound, the first isoester is the replace with the carboxyl group, right? Let me add a component here. Take the smiles. Oh, sorry, something else also happened here. I'm just taking this component, modified, and take the smiles of the acid replacement. Okay, right. Let me add one more cell here. I'm now calculating a properties for this modified compound. So, now number of heavy atom count is still 26. Number of donors are 3 now, and acceptors are 5 now, and molecular weight is 361, and log P is bit reduced, not completely. Okay, right? And if I take this compound to the here also, it's running, right? Okay, now let's see the aromatic nitro part. Just remember this, the positive impact, right? Now let's see. Can you see now? Previously, there is a red shade, right? That indicating, that can induce a toxicity. Now, the entire molecule is in a blue. Blue means a positive impact. There is no toxicity. So just by reducing a isoster, bioisosteric, when you say have a nitrogen, nitro group, so generally we used to replace by similar bioisosters, right? Now, can you see the impact now, right? So you don't need to address a molecule at one time. Every, replace the entire molecule with a replace this TPOC, replace this nitro, replace this aromatic nitro. So in a traditional approach, what we do, we try to address a one point at a time, replace one group, create a three molecules and see the impact on the target. Okay, so here what I'm trying to do is I don't want to use my knowledge. I don't want to spend time on the chemdra. Okay, let's break down this molecule and replace those the corresponding groups. Okay, so here I started analyzing the compound reaction scheme. This is my reaction scheme. The first one is addressing the POC protected amines, and next one followed by reductive amination. So this is a two-step approach. So don't worry about too much of Python code. So what I'm trying to do is, the first thing is I'm trying to take the list of amines from the existing vendors, okay, and trying to couple with here, okay, and trying to build the first component. And the next thing is trying to address the list of T-box, okay, and trying to do the radial amination. There's a two-step approach, okay, and I'm using RDKit, library emulation packages here. Okay, and the first approach is this is my input starting material. Okay, and the second step again reductive amination. Now the resulting compound you can see here, the first compound. Okay, now these are my the product from the first step. Now let's see the product from the first step before do initiating a second step, right? Now let's see after performing a second step. Can you see now you have a list of, instead of T-box tertiary butyl, you have a list of aromatic groups, right? And now let's see the properties here, right? Molecular weight versus density of the generated library, molecular weight versus log P, or molecular weight versus, different properties. Now the log P threshold, previously it was close to 2. Now I have many compounds in choice which are with a negative log P, which is very ideal for improving the solubility profile. You can see the distributions, right? Clear? So whereas here, this is my problem. So what I did was understand the molecule, understand the problematic areas, understand, try to improvise it. Either that can be done by using a regular manual methods, replacing one isostar at a time, and look for the starting material and either purchase the reagents and initiate the synthesis. So in addition to do that, so what I did was I retrieved a Molport catalog and retrieved all the acid chlorides or aldehyde that is required for my library enumeration, and I generated all the combination permutations, and this is my property distribution of resulting library. Now I can pick the compounds which are having a decent log P that will ultimately play a crucial role in my success rate of the improving the solubility, right? And toxicity, anyhow, I have already explored, and I was able to address those concerns also, right? So this is my library, and I can also add a filtration option to remove. Uh, I want to see the molecules which are having a less than 300 molecular weight, or acceptor, log P, or number of rotatable bonds, flexibility, number of aliphatic rings. I cannot filters, and I can see the best compounds which are filtered based on the physicochemical parameters. Then take the resulting library to the docking studies or virtual screening studies to understand their binding and their feature mapping. Then take the best compounds for the synthesis, right? So I did everything within the RTKit, within the Jupyter interface. I demonstrated that. Okay. I didn't use any other software to validate those analysis. Okay. Any questions? That's all I planned for today's event. Hope I was able to, demonstrate what I planned for today, and hope, the session was useful for you to start with the Articate and Jupyter Notebook.